Okay, we will go ahead and get started. Hello, everybody. Welcome to the very last day of the May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. Today is a very special day for us because our plan is to give this platform to our participants who were going to be attending this event in person to present their work. And we planned a very big event, a lot of social interactions and links, and so on, which unfortunately is difficult to replicate online, but despite all the adversity, we hope that we will have an opportunity to learn from each other and to understand how each of us approaches computational mass spectrometry-based research. Before we start, some logistics. So if you are joining us for the first time, at the bottom of your screen, you have two boxes. One is for chat and the other is for questions and answers. So please use chat if you have some logistics questions. For example, you cannot hear us, or the screen resolution is not good, or some other issue. That if you have questions to present, please use the Q&A button, because it allows us to handle questions more efficiently. Since we have a large group of presenters today, please indicate for each question the name of the presenter for whom you have a question. Because we will each presentation to 10 minutes, we will wait to ask the questions until the end of the presentation. And then if we have more questions that we can answer in the available time, we will take questions over to the end of the presentation, or we will make sure to answer them in writing. So this is the plan for today. Now, without further ado, let me go ahead and introduce our first speaker. So Chase from University of Illinois, Chicago. And Chase will be speaking about the fact that protein mass spectrometry is not only mass spectrometry. Programming in R for metabolomics mass spectrometry. And thank you for being part of this program.
All right, thank you, Olga and Mina, for organizing this. I'm really excited to get to talk to you today. And I'd like to say that I've also been following a lot of the developers that you're going to hear in the next talks as well. And I'm really excited to hear what they have to say as well. So this title is a little bit, well, first of all, I'm Chase Clark. I'm at the University of Illinois at Chicago in Dr. Brian Murphy's lab. And I will start with saying that this title may seem a little bit divisive. That was not my intent. I actually use both proteomics and metabolomics in my daily work at UIC. But when I was thinking about my journey into R and working on programming in mass spec, I thought about my frustration at the beginning and seeing a lot of proteomics packages, but maybe not as many metabolomics packages. And my background being in kind of biochemistry and protein chemistry, I was biased to thinking about proteins for mass spec. And my recent journey is more metabolomics. So I hate when people do this, but I'm gonna do it anyways. I'm gonna change my title to programming in R for mass omics, because I really think that using R for kind of any of the omics, metabolomics, proteomics, lipidomics, what have you, for mass spectrometry can be useful. And that also goes into kind of recent efforts in the R community. I'm not associated with this, but I wanted to highlight it. Things like R for mass spec, which are developing these low-level tools for working with mass spec data in R. So I really think that the community in R in omics is coming around to be able to support a lot of different applications in the mass spec community. My PhD is in pharmacognosy, and a lot of people probably haven't heard about that, but if you've ever seen Medicine Man, it's kind of like that. Unfortunately, our lab is not in the middle of the rainforest, but in Chicago. However, we do get to do field collections like Medicine Man. So pharmacognoscists are, in our lab especially, are really searching for new drugs from natural sources. So we go to places like Iceland and the Apostle Islands and all sorts of crazy places around the earth, trying to collect samples from weird locations and hoping that they have weird things. And we come back to the lab, and what we're really particularly interested in are bacteria that are living within any of these samples. These bacteria produce a diverse array of chemistry, and we test these for things like antibiotic activity. And then we try to figure out the chemicals that are responsible for this activity. And then in 10 to 20 years, maybe you have a new drug. But we had a problem when we came back to the lab, in that we didn't know which bacteria to study to prioritize for these further experiments. And when I started my PhD at UIC, Dr. Laura Sanchez also started her professorship there and brought some unique talents in MALDI-TOF mass spectrometry. And with this audience, I'm not going to go into how MALDI-TOF works, but I do want to highlight something important, and that's that we can collect 2 fingerprints for every colony with MALDI-TOF mass spec. So in a single experiment, we can get protein spectra, and these allow us to create these pseudo phylogenetic groupings of the colonies of the bacteria that tell us kind of who the bacteria are. In the same experiment, we can also collect small molecule spectra, and these are the measured, the produced metabolites that we're interested in. So when I started my PhD, before I'd even joined Brian's lab, he said, is there a way that we can join these 2 types of data together? And I maybe naively said, yes, I think that's possible. So I did a little research. There was a package that's was in well use, and I really still think it's a great package, called MALDI-Quant. And this was written in R by Dr. Gibb and Dr. Strimmer. And a few months before, actually, they had written this vignette on species identification with MALDI-Quant. But there was a problem. I didn't know how to code. So in my job previously, I'd worked with a software platform called Nime, which is kind of like Lego's coding. And so the first version of what I produced was in Nime, but it was really complicated to use. People had to install R, Nime, MS Convert, and get all those things working together. And it just really wasn't user-friendly. So I went back to the drawing board and thought about what was important for this analysis. And it's, can we go from colonies? Can we group using protein spectra? We know that this produces groupings that are similar to 16S gene sequencing clustering. And then also from metabolites, we're using these metabolite association networks where the large nodes represent isolates and the smaller nodes represent features. And we see these gross separations, but also the isolates and the features within the same plot. So this is really what we were targeting to do. So I set out to learn R, and right at the same time, Shiny version 1.0 came out. That allows us to build these kind of rich graphical user interfaces within R. Over the past few years, that's been iterated on a few times. And what it's landed on is IDBack is a package within R that was primarily focused around being built for a Shiny app. There's a whole nother talk about why this is good. One of the biggest benefits, I think, is you get the really robust testing and other organizational functions of a package in order to organize your Shiny app. And that's important when you get to a Shiny app that has around 200 functions and 5,000 to 10,000 lines of code. Another benefit of organizing a Shiny app as a package, in the case of IDBack, is that it becomes easy to script with it as well. And so the next version of IDBack is really streamlined in the ability to script with it and create really nice analyses, well, nice in my opinion, analyses in just a few lines of code. But originally it was a Shiny app, so I wanted to throw a few screenshots of the Shiny app in here. This is the landing page. You're able to compare protein spectra group protein spectra, as I showed before, but also plot any user-defined metadata in as well. And then also produce in real time these metabolite association networks that I talked about earlier by dragging over the dendrogram at any point. This is all pretty much built on top of and extending multi-quant and MZR packages, which are well established in the R mass spec community. But I also wanted to highlight that MALDI is a little bit different than like LCMS runs in that our sample number is really high. So a typical collection experiment will have thousands of isolates, so thousands of MZML files. And this is kind of untenable for a lot of labs to try to keep track of. So IDBack packages all of the MZML files and spectra and processed peaks into a single SQLite database for each experiment. So now instead of having to keep track of thousands of files, you can just quickly share a single SQLite database that has all of the necessary data. We've published a few publications on this. I'll share the slides for all of this afterwards. Encourage you to check it out, especially the Jove article if you want to learn more and see the Shiny app in action. Kind of the last part of this presentation, I just wanted to talk about how users access software, because this was a problem that I had to think about in developing IDBack. So for a simple Shiny app, you can do something like a web deployment on Shiny Apps IO if it's small enough, or the other typical way to deploy a Shiny app is to not deploy it at all, but just give users instructions on how to download and install it. These both have their issues. Web deployment can be really cost prohibitive if the files are large, like in mass spec, or long computation time. Local installation, a large percent of users aren't ever going to even attempt to install the package. So I came up with a few weekends of work. This package called Electric Shine allows you to create these installable Shiny apps with Electron that contain the R language and all the packages in your Shiny app required, self-contained within that installable executable. This works on Windows and Mac, and in the future, it will work with Linux. And I will point out that it's also compatible with continuous deployment. So I will highlight real quick that there's a lot of faces of mass spec software, GUIs, servers, and command line. With R, organizing it as a package in a Shiny app, you can pretty much hit all three of these with the same workflow without any duplicated effort. If you aren't familiar with creating Shiny apps as packages, there's a new package called Golem from GAR that I highly recommend for connecting these two. And with that, I will leave it there. I will want to thank the NIH for funding and all the group lab members for being great support during all of this.
Thank you very much. A big round of applause for you. And so we do have a question. Let me read it out. Is your SQLite backend read-only, or do you support writing, updating the underlying data?
Yeah, so it does support updating. It doesn't allow you right now to delete anything. I thought that was a little bit too dangerous. But yeah, there's interactive editing and adding of metadata, as well as appending new data into it.
This is great. Well, thank you so much. And let me remind everybody, please do ask questions or comments. You know, a big part of doing this person is that after the presentation, you can come up to the speaker and say, hey, I'm doing a similar thing. Can we compare notes? So it is more difficult to do this online, but please don't hesitate just to post comments or encouragements or anything that you would like to share, hopefully positive, with the speaker. So please don't hesitate to add things to Q&A. And even if we don't get to read them out, we'll certainly be communicated to the speaker after that. Thank you very much, Chase.
Thank you.
So our next speaker is Vlad Petyuk from Pacific Northwest National Laboratory in the US. And Vlad will speak about lessons learned from development of MSN ID package for handling MSMS identification. Welcome, Vlad.
All right, do you hear me? Do you see my screen?
We do, all good.
All right, perfect. All right, since Mina and Olga asked about not necessarily talking about package itself, but rather some tips, tricks that are during development. So here we go. My package, my talk, not necessarily about the package, but some tricks that hopefully can help others with the package development. All right. What was the motivation? Well, first of all, it's not a surprise to all of us that mass spectrometry data sets are huge. Computation is time consuming, and for a given project, we try different pipelines, different settings. So we run pipeline multiple times. Sometimes we run it, we exit the R session, then we run it again, then we again shut down laptop, then we run it again with another set. So there's a lot of repeated computation, and it can be a waste of time. Just happened a lot to me. So what are the options? Well, one is saving intermediate results into a file. That's fairly trivial. The other option is if we use our markdown to set cache equals true in chunk option. Next time we run it, the result is going to be out of cache. But what if we do it in a script or any other environment where we don't have that cache true option as in our markdown? So there's a technique for that. It's called minimization, and that's what I'm going to talk about. So what is exactly? Special type of cache technique, and the scope of minimization is a function call. Well, first of all, the function there shouldn't be any side effects. Otherwise, it's just not work. It's a popular technique in functional languages, where the inputs are values, the outputs are values, there are no side effects. Basically, it's implemented by checking, function checks if it's already pre-computed value for a given set of arguments. And if it's already has value for those arguments, it simply returns it out of cache without doing any computation. Function is not a trivial task. It sounds easy. I saw 2 and 2, and I need to add them. I return 4, but not really. If argument is a S4 object with the references and environments, then evaluation is not as trivial as it sounds. But nonetheless, there are two R packages that support memoization for your convenience: rcache and memoize, and they do a wonderful job. And hopefully, you'll find use for them in your future work. So let's start with an example, and this is just first classical example that in every textbook that covers memoization is computing factorial. So if n equals 0, by definition it's 1, else a return number by factorial of number less than itself by 1. So find conventional version, and then let's add memoized version. So, and we'll memoize this function by using memoize package. So let's compute memorial of 10. And I added a sleep just to make it a little bit slower, like there's some computing going on. So compute a factorial of 10. With first call, timing about 1 second in either case. Second time we call it, it's 1 second for conventional, and memoized rolls out of memory. So it doesn't do any computation. It can be also helpful if that call is hidden, like in this case, factorial of 11 is actually factorial of, if it's 11 multiplied by factorial of 10, so factorial of 11 is 1 second, and with memoized version, it's less than 1 second because it actually has hidden call to factorial of 10. All right, so we're done with classical example. Let's move on to a little bit more interesting example. Fibonacci sequence. So again, it starts with 2 ones, and then the next in the Fibonacci sequence is a sum of 2 previous ones. Like so, let's define conventional and memoize, and let's do first call Fibonacci of 10. And compute Fibonacci Fibonacci number. So in conventional case, it's 11 seconds, and in the moist function, it's 1 second. So what really happened here? We call this function for the first time, and it's already providing like 10x speed up. Well, it's actually not 10x. There's that. I can pause for a few seconds here so you can think about what really happened. And maybe it will be helpful if I show the previous slide. All right, and let's move on. So what really happened is that during computing Fibonacci sequence, there's a lot of wasted computation. So to compute Fibonacci of 10, you need Fibonacci 9 and Fibonacci of 8, and then so on. So for 8, you compute 7 and 6, and then for 9, you compute 7 and 6, and so on. So basically what memoization did in this case, it computed, it reduced the time complexities from exponential to the end to linear, because we need to compute all this only once, and we don't need to do this in duplicate, triplicate, and so on. All right, but that's all introductory computer science examples. Let's get to the real world, implementing in MSNID. And by the way, we don't really code with the recursive functions, also, although sometimes they're helpful, like inferring parsimony of proteins can be included with the recursive function, but it's not going to be efficient enough. But nonetheless, there's still roomization, I believe. One of the functions that I have in the package is called optimization of filter, of MSMS filter. If we have filter rock, then MSN identification, and we tell what's the maximum of DR we want to achieve, and we run optimization and optimizes the criteria that we define in filter object. And it's very computationally intensive. So I don't want to do it twice for the same object and the same criteria. And thus it's optimized with the memoization. So let's go through implementation. Well, first of all, since it's back to package S4, so here's generic. There's nothing unusual here. S4 method definition. In method, I call non-exported function, and that's the trick that Laurent taught me a while ago. It's just easy for debugging. And here's the definition of this memoized function. Yeah, the way it's called already memoized. Here's the moist function that simply memoized by adding rcache at memoization call. And here's the main definition of that function. So I hit a space by the package available on GitHub and by conductor. All right, so let's try. First, we read files for MSID object. And it reads MZID file, MZID files that stores MSM identifications. So first time it takes about, well, in this example that I used in this case, about 70 seconds to read those files. That's not a lot, but the example is also a kind of toy example, trivial. Then we define filtering criteria, and in this case, we want to use absolute BPM and spec value that's provided by MSGF search engine. Filter object will define it. So we want to retain less than 10 ppm, and those identification that have more than 7 of MSMS score, which was defined here as negative log 10. Show the filter object. Okay, here it is. So it's less than 10 ppm and more than 7 negative log 10 of spec value. Get filter on those PSMs, and for peptide identification, you have 10% FDR, which is high, the number of peptides, and accessions of proteins and peptide to spectrum matches. All right, so let's run optimization for first time. It takes 9 seconds, and here's the filtering criteria. Now it's less than 1 ppm, 7.7. Right, during these calls, what our cache did at the backend, it saves them as files hard drive. Next time we do the same calls, it takes only 1 second to read, less than 1 second to read files, and optimization also takes less than 1 second. All right, and sorry if I kind of ran really quick through the lessons because either of time limit. In summary, well, I want you everybody mindful that we're dealing with huge data sets, so please use techniques that can speed up computation, recomputation, and memoization is one of them for your service. There are 2 wonderful packages that support memoization in R, and I took advantage of that. So in case of memoize, it can store packages both in either RAM or hard drive for persistent storage. In our cache, it's a hard drive only, and I used R cache. So one caution rule: be careful about evaluating the arguments. It's not trivial as it seems, and I leave it exercise to the reader to look actually how those packages evaluate arguments for checking if they already computed values for those arguments or not. And thank you for your attention.
Thank you so very much. A big applause for you. So it is very interesting, and we have one question. Besides storage location, what are differences or advantages between r.cache and memoize?
When I was writing MSNID, R cache was the only available. Memoize is a letter package, and I haven't explored it yet, but one difference is that it can store calls in RAM, so access can be easier and faster, like for particular cases. I think for our goals, it doesn't really matter because we probably prefer to calls on hard drive, my guess, because of huge data sets. We don't want to really hog the memory.
Okay. Thank you so very much. So again, I do encourage you all to contact the speakers either versus Q&A or through other means. So good feedback that memoize seems useful to me, says one of the speakers. So this is good to know. So let us go on to the next presentation. So our next speaker is Timo Sachsenberg. I hope I pronounced right. I never actually learned to pronounce Timo's last name. University of Tübingen in Germany. And Timo is one of the developers of OpenMS, and he will talk about OpenMS from a developer's perspective. Thank you so much, Timo, for joining us.
Oh, thanks, Olga, for the invitation. Okay. So we all know that if we analyze one sample, this is usually not a big deal. We could even do it more or less manually. But if we analyze like hundreds or thousands of samples, this is really a challenge we are currently facing. And right from the start, when we start to develop OpenMS, we had the insight in mind that high throughput experiments require high throughput analysis. So ideally, such a workflow or solution would look something like this. You have your data. You have like a workflow or pipeline. You push a button and you get out the results that you can basically directly attach to your high impact publication. Unfortunately, this is not that easy. And as we all know, mass spectrometry is a very flexible bioanalytical technique. And that's also the reason why there are so many complications. There are a large range of different experiments. You can do proteomics, you can do metabolomics, interactomics, so on. There are different separation methods, acquisition strategies. You can do quantification, label-free, using labels, and so on, absolute quantification. And also there are like a lot of different experimental designs. So it's usually not possible to provide a single workflow or single tool that can solve all of these problems at once. And if there were, then it would probably look somehow like the Swiss Army knife on the slides. So a common solution is to provide a set of individual tools that, for example, because many of these analysis methods, they share some common steps or pre-processing or processing steps, and to just provide individual tools that can be combined in a flexible way. So we get like powerful workflows for specific analysis tasks. And there are several modular frameworks that exist. For example, Crux, OpenMS, but also TPP or Proteome Discover that follow this approach. But now let's get a bit into detail on OpenMS. So OpenMS is rather old, I would say. It's already has been developed for starting more than 10 years ago. We have more than 70 developers, including all kinds of students at university. And it's not a single tool, it's basically a framework for computational mass spectrometry. It's open source, so it's a BSD 3-clause license, if that tells you anything. So basically it means that it's free for academic and commercial use. And it's also available on the major platforms like Windows, Mac OS, and Linux. We exclusively use open standards like the HUPO PSI standard formats and have a quite large community distributed all over the world that contribute to OpenMS. And maybe one of the outstanding features of OpenMS is that it's designed to be scalable from single PCs, but also to high performance cloud or grid infrastructures. To give you an idea, so OpenMS did start as a C++ library that mainly only contains low-level processing algorithms. And building on that, more advanced algorithms and tools have been built. In total, there's about 180 application-specific tools, the so-called top tools. These are these Lego blocks you also heard before that can be combined in workflows. Then we also have PyOpenMS that interface to the C++ library, and it makes it much easier to, if you're not a hardcore C++ programmer, to use OpenMS algorithms. And then on top of that, we also have integration into different workflow engines. So for example, Nextflow or Galaxy, but also previously mentioned 9, as well as some commercial software. So you can do basically very low-level processing of your data, where you get the raw performance of C++. You can use tools that you combine to create customized workflows. And you can also have like workflows that do one specific analysis. As you can imagine, maintaining the development process on all these levels is kind of challenging. And so I want to give some advice. I mean, maybe for some, everything that's already clear and doing that, but maybe for some, it might be good feedback from a developer perspective. So the one important thing is that to maintain such a large and diverse project is to establish best practices. This means you need to have proper source code control, version control on GitHub in our case. We perform extensive automated testing. And the nice thing is that in recent years, this changed dramatically, and you can do this very easily on GitHub, both version control and testing your software. A lot of different providers are out there, like Travis or CircleCI, GitHub Actions, and so on. And the other important idea is that you provide continuous integration. This means you only allow reviewed and working contributions into your code base, because changing a little thing at one part can break like maybe the Python bindings on the other part or some workflow. And also you get some testing on multiple environments and platforms. The third point is continuous deployment, that you only provide working installers and containers to the public, maybe like in a Nike build. And I think this is so universal that it doesn't apply to C++ or OpenMF. I think like more or less every project should follow these principles. This is one example. Somebody opened a pull request on GitHub for OpenMF. And you can see here a lot of tests have been run, different platforms, Windows, Linux, Mac OS, and all of them passed except one, the one that tests the integration into PyOpenMF. And this, in this case, would have never been caught if you just developed a small change on your computer, probably never test specifically the Python integration. So what is also a challenge? In general, it's dependency management if you do more complex workflows, not only a single tool. Some of them can be managed by the workflow system, for example, KNIME, Galaxy, or PD. Basically, everything comes included. Or you need to rely on external dependency management. So you could ask, if I have like multiple tools, search engines, and I have a workflow, I could just create a container, install all dependencies manually in it, and it will run. Yes, that's true, but I would suggest not to do that. There are better solutions to that. And maybe you might take a look at Bioconda if you don't know that yet. It's also hosted on GitHub. And there are so-called recipes that specifically describe how your container is built. And the recipes describe how the container is built. And this allows you to generate so-called biocontainer in a reproducible way. So you track all version numbers of your dependencies, and also you are able to generate containers, maybe not only for Docker, but also for Singularity and other container engines. And this allows you to limit the effect of installation and execution of tools and enormously improve the reproducibility of your results if you do that. It also is often necessary that you containerize your software to run it on the cloud or HPC infrastructure. And again, the question is, how can you make sure that these containers that you publish always work, especially if many people work on them? And the answer is, again, continuous integration. So you need to test things. If you now have such your workflow running and your tools work well, you should also promote them. So people learn about them. Sure, you can publish, but you should also make them accessible via multiple channels, like source packages, via package managers, installers, containers, and so on. You can register them. There are some places like the Galaxy Toolchat or KNIME Hub, where you can register your workflow, or can also include details in BioTools or Docker. And as always, it's nice to provide example workflows. The last part I wanted to talk about is how to make your algorithms more accessible. I think the people in the R world are pretty good. You have a very standardized way how to provide your algorithms. In C++, this is totally not the case. And also, in our experience, it's usually hard to get students or others that are really experts in C++. So it's really non-trivial to learn. Sometimes it's really pain. You also always do the compiling. This takes ages. The dependency management in C++ is awful. So at some point, we decided to provide the Python bindings because Python is easier to learn. it's mature. You don't have a compilation step. You can develop much faster, and you also can easily install other scientific libraries, as well as like an ecosystem of Python mass spec libraries out there. So what we do is we construct, every night we create a Python wheel that takes basically our C++ code and some description that tells what should be exported to Python. And the so-called the auto-wrap tool uses Python to map, basically create Python classes that then call the OpenMS classes and data structures. And it's so far we wrapped several thousand of the C++ function classes, including many of the file formats that might be interesting for you. One nice use case, so there are several nice use cases of PyOpenMS. One of the things is that you really can quickly prototype stuff. So if you need to publish something quickly, there's a new experimental method out there. It's usually rather convenient to prototype your method in PyOpenMS. Also, we have some basic support to create graphical user interfaces. And as mentioned before, you can also integrate with other packages like, for example, PyMol. Recently, we started to look at R integration via Python. So there's a so-called reticulate package. And maybe next time I can talk about that. So, yeah, you will find OpenMS on our GitHub page. And yeah, can install PyOpenMS via Bioconda or pip.
Thank you so much, Timo. Thanks a lot. So let's see if we have any questions. Let's just give it a minute or so. And if we don't have questions now, we can certainly have questions later. So I think Timo has such an extensive experience, and the whole OpenMS team has such an extensive experience in developing modular, reusable, sustainable software that I think we can learn a lot from this experience. So I'm really grateful that you could present today. Thank you so much, Timo.
Thanks.
So since we don't have any questions right now, so let's just go on right to the next speaker. So our next speaker is Shubham Gupta from University of Toronto. And Shubham is a member of the OpenFlat team. And we're also very happy to have this opportunity to hear from him. And so the topic is automated workflow for peptide level quantification for DIA-MS and SWAS-MS data. Perfect.
Hi, can you guys hear me?
We cannot see you, but we can hear you if you would like to turn on your video.
I think I did. Oh, 1 second. Okay, so.
Oh, you are. Oh, good. Yes.
So yeah, I'm presenting here the automated workflow for DIA data analysis. And so why do we need first the automated workflow? First of all, like you can look at it like the DIA data is very complicated. It's very convoluted spectra. It's really hard to analyze kind of data manually. So you need like good softwares to find the quantitation of your analytes. So first of all, like this is a high throughput data, and also like if you do it like manual analysis, you will introduce some like biasness. So that's why we need some unbiased analysis for it. So in our lab, we are focusing mostly on the open source softwares. And why do we need open source softwares? First of all, they're affordable. You can like get them for free. Second of all, and very important, is transparency. So with as Timo presented, there were like OpenMS software developed, and like all the code is available. So when you analyze your data, you can really go through the code as well if you want to, and you can understand what's really happening under the hood. The third part is interoperability. So I kind of copied some of the script from OpenMS for my purpose. So it kind of makes downstream tools to be compatible and like to be consistent for like consistent analysis. And third part is reproducibility. Fourth part, so we kind of use Docker to get the reproducible analysis from our pipeline. So in my work, we are focusing on 3 softwares: OpenFlat, PyProfit, and DIA-LAN-R. So you get the DIA data. I think you might have like gone through many tutorials for learning DIA. So first of all, like you find the chromatograms and you score them, like score the peaks in the chromatogram. In the PyProfit, you generally get the false discovery estimation for all the peak scores, peak you found, and then you select the best for the quantitation, and then we do the alignment for aligning the low intensity peaks that were not identified before and kind of map the peak boundaries or find the features that had low scores. So this was good to know about. However, it's like really difficult to do analysis because it's really hard to install the software. So it's kind of very painful. So to ease the installation process, we kind of focus on using Docker container. What it does, you can download the image easily, and also it kind of produces reproducibility. So with the Docker, you have this image, and then they have like the final tag here. So in this case, you have the version number that tells you that what kind of version did you download. So if you download the same version and run the analysis, you should get the same output. For the DIA-LAN-R, we have the package on the bioconductor, so you can do the analysis in the same way. So let's go to the DIA part. So in DIA, mostly we have peptide coming out from the column, and then we have MS-MS spectra. So where peptides gets fragmented into B and Y ions. And if you look at the MS2 spectra and the transition mass to charge, if you extract the transition mass to charge signal, you can recreate this kind of chromatographic illusion of the peptide. And if you find this co-illusion, you say this is my peptide identified, and area under the curve would be the quantification. So with OpenFlat, it's a targeted way of analyzing data. So you have an assay library that have like all you call the peptide, and then the spectral library. So first of all, OpenFlat kind of aligns the spectral library to the assay library using either the spiked in standards, that's like IRT peptides, or some endogenous high intensity peptides. So once the library and the sample is aligned, it kind of extracts chromatogram. So for each analyte or each peptide, it gets the transition mass to charge and the retention time, and then extracts like plus minus 5 minutes because you don't really know where the peptide will elute the chromatography. So around the library time, you extract plus minus 5 minutes, and then you kind of get this chromatographic trace. So in this case, you have so many peaks identified, and now you need to find out what's the correct peak group. So OpenFlat creates multiple scores for these peak groups with respect to the library and with respect to the traces itself. Then we use PyProfit. PyProfit then kind of do the false discovery rate estimation and get the p-value or q-value for, so it gets a p-value from those scores, and then it do the FDR and gets a q-value. So you get this lowest q-value, and then you say this is my correct peptide. So this is work fine for like high intensity peptides, but for low intensity, sometimes you may not identify the peaks. So for that, you need some alignment. So there are multiple alignment strategies in the literature. The first one is very simple linear IRT alignment, and it's very simple. You can just find the IRT peptides and align one run to another run. The second little bit complicated is nonlinear. So we have this low S strategy, and the other software tools as well, like Trade, kind of find the nonlinear pattern between two runs. However, the problem with these two methods is they are all global, and like they kind of have a monotonic alignment. So we developed a new method called DIL&R that kind of does a global plus local. So it kind of finds the alignment for each peptide separately, with irrespective of other analytes. So we did it on a heterogeneous data set, and we found that with IRT you only get 62% alignment, with global nonlinear you get 76% alignment, and DIL&R you can identify like 98% alignment. So it's really state of the art in terms of the DIU data alignment. So the way it works is, first you find the reference run for each analyte, and then you find the reference run for each analyte, then you align the chromatogram for one run to another run. And with the alignment, you can really map the peptides from one run to another run. So in this case, this peptide could have been missing because of the low intensity, but with this alignment, you can find the quantitation. So the way the alignment works is, first you have this chromatogram from one sample and this another sample, and you kind of build this similarity matrix that's point-wise similarity matrix for each point between each time point for the alignment between two runs. And then we do the dynamic programming to further find the alignment path. The problem with this whole strategy is this is in terms of like n squared, because the computational complexity of this analysis, it grows very exponentially with the length of the chromatogram that you choose. And and we found out this is the bottleneck of our analysis pipeline. So to solve this problem, we used package RCPP. So this is this package allows to connect C++ code with the R code. And what we found out that with this strategy, we were able to get 35 times faster analysis time. So for 10,000 peptides, in R, it would take 20 minutes. And if you use the C++ code for the alignment, it would take 35 seconds. And so what it does, it kind of enables the whole analysis to be done on a single laptop, and you don't have to use the computing clusters. In addition, like we kind of then interactively visualize all the alignment, because there's a very little latency now. So we have this paper where we have the RSANI app for the visualization of the alignment. Another thing we did with our package is we used the OpenMS C++ scripts for the peak integration. So when we map the peak boundaries, it kind of, we find the quantitation in the consistent way, the way we have done in the OpenSWAT. So these are the two benefits using the RCPP in the R package. So the current structure looks like this. We have the computationally complex part done in C++, and then we have this MZR package for reading the chromatograms. That's the output from the OpenSWAT, and then we SQLite package to read the SQLite file from the PyProfit output. And with this allows us to use even the Python on top of the C++ code. So you can, with this alignment algorithm now in C++, you can use a Python binding or Ruby binding or any other things on top of it. And what it does is like kind of, it kind of speeds up the whole calculation and make it useful for other software tools as well. And the regular chromatogram extraction, smoothing, finding features, getting the final output, it's all done in regular R. So with this thing, we have all this, this tutorial we have compiled in a document, in a book chapter, and it's available. So you can, if you want to use the whole workflow, you can easily do it by reading the instructions from here. And with that, I'm very thankful to Professor host.
Okay, thank you so very much. Thank you for this presentation. We do have one question to you. Does this tool also support MSE data from waters?
Oh, so right now we kind of read the chromatograms from the OpenFlat. So OpenFlat outputs the chromatogram and this kind of supports the thing. So I'm not really sure if OpenFlat software supports H2E data set. So if it does support it, then it would probably work.
Awesome work. Thank you for the feedback. And one more question. So that was one of the participants writing. And one more question. Is retention time alignment sensitive to the retention time window size? If yes, is there any method to find the optimal size?
Yes, so retention time alignment is sensitive. So if you have wider window size, means you have many other peaks. So with this alignment thing, we are kind of mixing global approach plus the local approach. So local alignment would be if you have only a small window size around the peak, so it would be perfect for the local alignment. But to increase the window size, then there would be more peak, so it would tend to go towards the global approach. So we don't really know right now what's the optimal window size is, but generally we follow the OpenFlat strategy that's plus minus 5 minutes to expect our main peak of interest to be within that plus minus 5 minute window. So that's what we use.
One more feedback from a participant and a question. First of all, thank you. And second, will this alignment also become available in Skyline at some point?
We have a C++ code that have does the alignment. So in principle, it should be easy to integrate into the Skyline. However, we haven't done that part yet. So it requires some more work to kind of bind this alignment algorithm with the C++ work, with the Skyline.
Okay, thank you very much. So I don't see any more questions. So let us move on to the next presentation. So our next speaker is Michał Ciach from University of Warsaw in Poland. And Michał will speak about Messerstein, a robust approach to linear deconvolution using optimal transport.
Hello, everyone. Can you hear me?
Yes, and
Great.
see your screen.
Thank you for the introduction. Okay, so the linear deconvolution is a way of estimating the relative abundances of molecules in a mass spectrum. Let's suppose we have an experimentally measured mass spectrum here, and we want to estimate the quantities of a set of theoretical isotopic envelopes like these ones. In linear deconvolution, we express the observed spectrum as a linear combination of the theoretical spectra plus some additional noise or additional signal coming from other molecules. This approach is ubiquitous in all fields of mass spectrometry. For example, in resolving isobaric interferences, where we want to quantify molecules which have the same nominal mass. In assessing the polymerization efficiency, where we study the chain lengths of polymers. In processing the data-independent acquisition label-free quantification experiments, or in annotating mass spectrometric images. The way it's most commonly done is by some sort of linear regression in which we first centroid the experimental spectrum and then treat all the spectra as vectors of intensities corresponding to different masses. And there is a whole list of problems connected with this approach. One of the major ones is that the measured and the theoretical peaks simply do not match. There is a naturally occurring variability in our measurement, which is then even inflated by peak picking procedures. Peak picking of or centroiding of complex spectra is a difficult task. And the methods are either computationally expensive or inaccurate. And one of the ways this is usually circumvented is by setting some threshold such that when peaks are closer to each other than this threshold, they are treated as one. But this introduces further problems. And we wanted to take a different approach. We wanted to take a different definition of what it means that spectra are similar. And for that, we used the Wasserstein distance based on the theory of optimal transport. The Wasserstein distance between a pair of spectra is the optimal cost of transport of the signal from one spectrum onto another. And this is best explained by an example like this one. We have two spectra overlaid here, an orange one and a blue one. And we will transport the signal from the orange spectrum onto the blue one. We start by transporting 1/3 of the orange intensity from the leftmost orange peak by 2 daltons onto the first blue peak. The remaining intensity from this peak gets transported onto the next blue peak. This transport gives us partial costs. The first was 2/3 of a dalton, and the next one was slightly larger. And the first peak is now fully transported. So we move on to the next orange peak. It gets transported onto the blue peak at 111 daltons, and we are left with a small remaining peak at 120. This peak is far away from the nearest blue peak, but because it's a small peak, it only contributes a little bit to the final value of the distance, which now is equal to 4.6 dalton. And in this way, the Wasserstein distance accurately measures the similarity between spectra in both domains, both in mass and the intensity domain. Two spectra are more similar to each other the closer their peaks are positioned, and the more similar their intensities are. And in this way, this distance is naturally robust to small measurement errors. The mathematical definition is a bit more complex, but the nice thing about this distance is that there is an algorithm that allows us to compute it in a linear time and memory complexity. So from a computational point of view, it is not more costly than, for example, the spectral contrast angle. The linear deconvolution using the Wasserstein distance now boils down to finding a set of proportions that minimize this distance between the experimental spectrum and a linear combination of the theoretical ones. We couple this with a simultaneous removal of excess signal from the experimental spectrum to accommodate the presence of unexpected molecules. And this computational problem can be transformed into a computational optimization of a linear function with respect to, say, set of linear constraints. So the point is that the key point here is that even though this distance might seem quite complicated, we can actually, we have quite efficient algorithms that allow us to use it to estimate proportions of theoretical spectra. We illustrate the capabilities of this method with a set of case studies, one of which is studying the phospholipids in a tissue section of a mouse bladder. We've downloaded a dataset from the Pride database, and we have deconvolved each pixel with respect to a selected lipid, formulas. On the left-hand side, you can see an optical image of this tissue section. And on the lower bottom, on the bottom part, in the results of a small case study in which we analyzed 3 formulas which were previously studied in this data set. Now, linear reconvolution allowed us to analyze the spatial distribution of those lipids in a more robust way than, for example, analyzing the intensity of the monoisotopic peak, which was especially important for the lipid PE38-1, in which case we had an interference which highly inflated its intensity estimated from the monoisotopic peak only. Perhaps a more interesting case study was a larger one in which we deconvolved each of the almost 35,000 pixels with respect to 2,500 formulas. This has allowed us to perform principal component analysis in the space of metabolites instead of the space of peaks, which allowed us to better identify and characterize different tissues. We have implemented our methods in a Python 3 package called Wasserstein, mass spectrum analysis using the Wasserstein distance, which is available freely on GitHub. And I would like to thank you, and I'll be happy to take any questions.
Thank you so very much for the fantastic presentation. I'm particularly interested in seeing the data set which Kylie presented yesterday as well. So it is really nice to see the intersection. So let's see if we have questions. Maybe let's take a minute or so. And if we don't have questions now, I hope that some of you will have questions for later, because I think that this is definitely a very useful work that can be used in many, as you said, in many different types of applications. Okay, one feedback is very nice talk. Another participant says very nice indeed. So a lot of encouragement. Looks like no questions so far, but a lot of positive feedback. So, you know, I have a feeling that you will be getting questions later. So, okay.
Well, and thank you very much.
And thank you to the participants for encouraging our speakers. Please send feedback even if you don't have questions. This is a really good thing to do. So let us move on to the next speaker. So our next speaker is Melanie Odenkirk from North Carolina State University in the US. And Melanie will speak about structural-based connectivity and omic phenotype evaluations for lipid data interpretation. Welcome, Melanie. We look forward to hearing about your work.
Thank you, Olga, and thank you all for virtually tuning in. My name is Melanie Odenkirk, and I am a grad student at Aaron Baker's lab in North Carolina State University. Like Olga said, I'm going to be telling you about a workflow we have built to improve lipidomic data interpretation capabilities. The full name is a structural-based connectivity and omic phenotype evaluations workflow, but we typically just call this SCOPE. Before I get into talking about SCOPE and its capabilities, I want to start talking about what motivated us to build SCOPE in the first place. And a lot of this stems from clinical research questions and trying to better understand why a patient's getting sick, how to treat patients, and detecting diseases before a person has symptoms presenting. And to do this, we follow this very general workflow on the bottom here, where we'll first design an experiment with a particular disease in mind. We'll then collect data and then do any kind of data analysis, such as processing and statistics, and then we move into data interpretation. I think a lot of people have data analysis and data interpretation as being very synonymous terms to one another, but I like to think of these as very separate steps because to me, data interpretation is taking our statistically significant molecules and relating them to their biological implications, whether this be through processes or pathway annotations. This is a really important step in this workflow because it's ultimately going to determine the conclusions we can make and the answers we give for these clinical research questions. And so while omics like proteins have extensive databases such as UniProt and KEGG for this biological interpretation step, other omics like lipids face greater challenges. And so I just want to give you an example of what I mean here and why biological interpretations of lipids struggle. So up top here, we have two lipid structures. On the left, we have a PC 180181, and on the right, we have a PC 140226. If you're unfamiliar with lipids, hopefully you know that they have a polar head group and non-polar fatty acyl tails. And that's what we see for both of these lipid structures. In our green circle is that head group annotation. Both of these lipids have the same functionalization, and they're both PC or phosphatidylcholine lipids. Where we see a lot of structural variation between these two lipid species is in their fatty acyl tails. So typically, we can count the total number of carbons and also the double bond information, which is given before and after the colon in each of these lipid names here. So the problem with interpreting lipids on a biological scale is often we're really left to compress down these lipids, which you can see here are quite structurally different from one another by their head group annotation, as shown in the middle right of this pathway here with this PC node. This becomes problematic when we have both down and up regulation of lipid species within the same node of this pathway. So if this PC 180181 is down-regulated and this PC 140226 is up-regulated, having both this up and down regulation in the same node of this pathway makes understanding our system on like an entire level very difficult to do. So our goal with SCOPE was to provide a platform to more comprehensively answer these clinical research questions. And so the first way we want to kind of improve upon what exists now for lipids is through this structure-based connectivity. The idea that structure is related to biological function is by no means a new concept. And we talked about that head group and that fatty acyl information before. Lipid head group is actually really important for determining our lipids biological role. And so we have this PC species again, but we can have quite a bit of structural differentiation within lipids that we might detect in human samples. Just as an example, these two other head groups on the bottom here. And that fatty acyl information is also really important for our lipids enzymatic specificity. The other area we really want to improve upon and expand is this omic phenotype evaluation. And essentially what this is boiling down to is trying to better understand clinical variables and how they might affect biological variability we observe in a study. One area for lipids we might be particularly interested in is diet, because lipids are really integral parts of foods we eat every day. And this can result in quite a bit of fluctuation in abundance for lipids we can detect with mass spectrometry with patients in the same disease or a healthy group. And so we're not just talking about diet as being one of these confounding factors. We could also have things like age, sex, and medication that can also really influence our entire experimental setup, from everything from that experimental design stage we start out with to testing lipids as biomarkers for a really robust population. And so our goal with SCOPE was to take this biological dysregulation we might observe and to relate both this structural annotation and these metadata effects as well. For the sake of time, I'm just going to take you through a few highlights of how we've built these associations. And so the first thing I want to talk about is doing structural connectivity with a lipid head group. And so if we just take these four lipids here as an example, a ceramide 181241 up top, two PCs in the middle here, and then a PE 160181 on the bottom. We want to relate these species by their structural similarity to get at this head group association. And so we do this in a hierarchical clustering technique where we can relate species by their structural similarity, which is ultimately going to be driven by different functionalization of our lipid species, which tends to happen in this head group region. And so if we go to relate these species by their structural similarity, the two that are going to be the most similar are going to be our PCs here. And so they would cluster together first in a dendrogram-like structure. Our next most removed would be our PE on the bottom here. And then finally, the ceramide up top would be the furthest away because it has the greatest structural differentiation. And so if we imagine doing this for all lipids we detect in a biological sample, we can build this dendrogram that's going to show how these groups were combined. And again, this effect is primarily driven by our head group annotation. And so just to give you an example of what this looks like, this is a human plasma study where we identified 288 unique lipids. So here, every node on the end of this dendrogram corresponds to one of those unique lipid identifications, and we're just coloring by that head group annotation. What's really nice about this technique is we're still able to build those head group visualizations like we do typically with lipid pathways, but we're still keeping all of these lipid identifications as a unique species here. And so what we can then do is overlay results from our statistical analysis. And so here I'm just showing you the log2 fold changes we observe for all of these lipids for our disease versus control comparison of this example data set. Anywhere you see gray, our species was not statistically significant. Red is going to be upregulated or higher in our disease group, and then blue is downregulated. We also use a color gradient to show the magnitude of the log2 fold change we're observing here, with a darker color indicating a larger value. And then so the other part of this workflow that we've been working on is mapping this clinical data or metadata effect. I'm just going to show you how we've been doing this for individual lipid species and relating lipid structure to this clinical information. And we do this using a software called ToxPy. This software was originally developed at the EPA, and we've since adapted its more environmental application to better visualizing these clinical data effects. So to do this, we'll take all of our patients and we'll first group them by a specific clinical variable. So here we're grouping by age in increments of 5 years. Then for every single unique lipid identification we have, we'll generate a ToxPy that looks very similar to one in the bottom left here for this PC16018.3. Here, the height of the slice corresponds to our log2 average abundance within each of these patient groups, and the width of the slice corresponds to how many patients are actually within each of these disease groups. And so as we go around from our dark blue to our light blue to our green here, our age is increasing, and this lipid appears to be very subtly growing in as we get older. So in conclusion, through SCOPE, we can take this biological dysregulation we might observe and use things like analyte structure and also this clinical data to better understand some of the biological dysregulation we observe to ultimately better answer clinical research questions. There are a lot of people who have contributed to this work, so I want to give them a shout out. But the 3 people I want to thank the most are my mentor, Aaron Baker, and 2 of our collaborators, Denny Forsch and David Reif, also at NC State, who have really made this a team effort. Thank you all for listening.
Thank you so very much, Melanie. Let's see if we have any questions. So we don't have any questions so far. We'll just give a couple moments if someone has a question. This is definitely challenging work and very important. So it is very nice. So I think we do have one comment saying for one of the attendees, great presentation, very clear. Thank you very much. Yes, thank you indeed.
Thank you.
Okay, so in this case, let us go on. Oh, let's see one more. Good talk, thank you. So here you go. Thank you very much, and thank you for the feedback to everybody. So let us move on to the next talk. So this is the talk by Alan Hubbard from Donald Danforth's Plant Science Center in St. Louis. And Alan will speak about using untargeted metabolomics and genome-wide association studies to identify conserved networks in the C4 grasses.
Yeah, great. Let me just get this set up. Okay, so I'm going to transition a bit to start talking about how we're actually using these technologies in the plant sciences. And I think this dovetails off of some of the themes a lot of the speakers have been talking about. So in particular, in the lab that I work in, we want to use metabolomics to understand the drought stress response. And a 10,000-foot view, why do we care about the drought stress response in plants? Well, we know that drought stress is bad right now, and it's going to get worse in the future. And so it makes sense now to start planning ahead about how are we going to adapt plant biochemistry to a changing climate where drought is far more prevalent. So it's a big agricultural priority, but there's something that's really held us back from leveraging the really cool technologies that are emerging in plant genetic engineering, plant breeding, to actually change biochemistry. And the fundamental issue is that it's very difficult right now, and it's been essentially impossible to quantify all of the the chemicals that are in a plant in the first place. So how can we actually track those chemicals as they change under drought stress? And so a lot of plant scientists are now, super attracted to this idea of taking a bird's eye view of biochemistry and are drawn to untargeted metabolomics because it provides the avenue through which we can really get a holistic perspective of all the biochemical systems that are changing under drought stress. And in the lab that I work in as a postdoc, Ivan Baxter's lab, we're particularly interested in exploiting untargeted metabolomics to do comparative genomics. And because untargeted metabolomics allows us to track the full complement of biochemistry changing under drought stress in a single plant species, we've decided to expand this approach out and to actually compare how the metabolome of a model species Ceteria compares to a crop species sorghum, as both are exposed to drought stress. This enables a lot of really deep biological questions because we can actually understand at this point how deep mechanisms of regulation are conserved at the metabolic biochemistry level. However, to actually answer these really cool biological questions, to see, what biochemical regulation is preserved under millions of years of evolution, we need lots and lots of samples of Ceteria and sorghum. And so to actually implement this experiment, we're going to be doing untargeted metabolomics on 1,800 Ceteria samples and 1,800 sorghum samples. We're going to drought stress half of them, and we're going to look across hundreds of genotypes in each species. So it's a massive amount of data set to do untargeted metabolomics. How are we doing untargeted metabolomics? Well, this is what brings me to a mass spectrometry conference. It turns out that mass spectrometry is cheap and it's reliable, and it provides, in theory, all of the information that we need to separate out metabolites in all the samples we look at. We can use retention time to separate out by structural properties, and then the mass to separate out by mass, and the spectrometer component is going to get us very thin retention time windows so that we can get very high quality signals for a number of metabolites. And in theory, the combination of knowing the mass and retention time should allow us to detect, each of the individual metabolites in our 3,600 samples. But I want to emphasize that there's a huge gap between theory and reality. And so, although it makes sense that mass and retention time is enough to distinguish individual metabolites from one another so that we can get their measurements of relative abundance by intensity, in actuality, when we look at metabolomics data, due to the natural variation of using mass spectrometry, we see a lot of variation in intensities, not just intensities, but also mass itself and retention time due to the intrinsic error of using a column and the orbitrap that we use. And so your actual landscape of data is more of this sort of distributed point cloud from which we need to find a way to map back this very noisy data to the clean, discrete categories that represent individual metabolites. And I need to emphasize that to actually answer biological questions, we are doing this on the scale of thousands of samples. And so how do I actually use existing informatics technologies to do this? I've put together, working in collaboration with the Douglas Allen Lab at the Danforth, I think a really nice pipeline to actually call accurate metabolites from this very noisy data set. We use a software called isotopolog optimization, which tracks very high confidence peaks, which are metabolite peaks accompanied with very prevalent and detectable isotopologs across samples to sort of monitor experimental noise that can be corrected for when we calculate noise parameters. I use a separate software called XCMS to actually call peaks using the noise parameters calculated from IPO. And then we prune out from this, the hundreds of compounds that we get, any of the possible salts using a salt filtering pipeline. And so using basically, off-the-shelf open software, we can put together a really good pipeline to let us call metabolites in theory across these hundreds of samples. But we need to keep in mind that we're going to not only need to call signals, but we need a way to ground truth them. Because the data is so noisy, how do we know that the signals that we think are metabolites aren't just spurious representations of noise? And so what I've been thinking about and we've been working with in collaboration with other labs at the Danforth is effective mass spectrometry workflows where we can monitor and control for noise in these untargeted approaches. And what we found is probably the best way to do this is to use spike-ins to monitor experimental noise, just intrinsic to mass spectrometry. So the concept of a spike-in is fairly straightforward. So let's say that I was to see a very high confidence signal of something I would expect to find in my plant samples. Let's say I was looking at fructose. I would see a signal similar to what we see in this yellow box, a very high confidence, high intensity, major signal accompanied by potentially 2 isotopologs. And I would see, hopefully in every sample, a good fructose signal, but maybe there's a lot of variation from sample to sample. How do I actually know that that variation is potentially biological noise or technical noise? Well, one way to do that would be to add into my sample perhaps a spike in of 13C labeled fructose. This fructose that is labeled with 13C is going to have a different mass. It's going to be at a fixed concentration. So I'm going to have a totally independent signal with the same chemical properties as my endogenous fructose that will provide me sort of a benchmark from which I can normalize and I can monitor experimental noise. So standards are great, and it's a really good recommended element of experimental design in untargeted approaches. But working with plants, with hundreds of thousands of metabolites, I face this sort of unique predicament is how do I actually choose my spike-in? I'm not just looking at one chemical such as fructose, I'm looking at the full complement of hundreds to thousands of metabolites. How do I actually have enough spike-ins to get me the full range of different compound classes? And the solution to that is something that we've been pushing forward for the plant science community, is to start using an unbiased set of standards from a 13C labeled metabolome of yeast, which you can get by simply growing yeast on a 13C media purchased through Cambridge Isotopes. And this approach works really well because we can look at the chromatograms and the overlap with isotopic masses and see that because yeast and plants are both eukaryotic organisms, there's actually a very good degree of overlap in the metabolites found in each. And we know already thousands of well-characterized metabolites in yeast that we match up based off of their masses and retention times. And so what we're able to do is by looking at the overlap between the 13C labeled yeast and in our plant samples, we can find at least dozens to hundreds of metabolites for which we have a good endogenous signal in plants and also have a potential corresponding standard in yeast. So it looks like that this is really a sort of gold standard for having a way to, in a totally unbiased, untargeted fashion, find very good standards that are going to go across the beginning of your mass window all the way to the end and across many different retention times. There's a fundamental sort of roadblock that we hit when using this approach though, and it is that the cost is somewhat prohibitive to use yeast standards at the suggested 1:10 dilution. And so what we've done is experimented with different dilutions and expanded beyond the several compounds that were recommended to us to look at by the Cambridge Isotopes company as we've looked at the full complement of the yeast metabolome. And I wanted to walk through the results of some of these experiments to show that we can get away with a much higher dilution and still capture all the relevant information of sample-to-sample variation. So we have dozens of compounds that look to be very high quality in terms of both getting a good plant signal and a 13C yeast spike-in signal. So what we're looking at here is one such compound, citrulline. On the left, we see our endogenous plant version in sorghum extract, and then we see on the right the 13C version, which is just the compound that's found in the yeast 13C labeled extract that has been added to our sorghum sample. And so what we see from this, the take home, is basically that there's a lot of sample-to-sample variation looking across 3 technical reps and 3 biological and 3 technical reps across these 9 samples. We see substantial variations in peak intensity. And importantly, this peak intensity due to technical noise is replicated very effectively in our 13C labeled standard. So the variation in our endogenous compounds is reflected in the standard. And this is really important because we're going to be limited to by technical variations when we do larger sample sizes. So we see that at a 1 to 20 dilution, we can effectively track the landscape of variation due to technical noise. And I had mentioned that cost is prohibitive. So, already getting up to 1 to 20, what we see is we can increase the dilution further to 1 to 50. We think this is probably the max for some compounds, but when we increase to 1 to 50 dilution, it's a little bit questionable, the signal that we get for some of the samples, but we can effectively still track the landscape of variation for citrulline and many other compounds. And so what we've been able to see as we've developed our workflow for LCMS, moving from having informatics technique and then being able to monitor how well that performs through yeast spike-in standards, what we've seen is that probably the best set of standards is not a targeted approach, but it's this untargeted inclusion of a 13C labeled yeast metabolome. And probably at one of the higher dilutions, 1 to 30, potentially 1 to 50, which is very important because it brings the cost of this technology down to the point that you can do hundreds of samples per vial. And what you're going to get out of spending that money of resources is a set of standards that will give you a way to track variation across different compound classes, since the yeast metabolome contains lipids, sugars, amino acids, and the variety of other things you would also find in plant samples. And so I wanted to just sort of give an update of where we are with this right now, because my abstract had been focused more on presenting the results of potential metabolites we had zeroed in on, but we've been held up by the pandemic. So what we've done is finalize this workflow and chosen more standards from the yeast, sorghum, and Soteria samples, so that we can really accurately monitor technical and biological noise. And we're working on writing this up into a methods that's really going to make quantitative genetics across species using LCMS possible. And as soon as our facilities open up, we're going to implement this methods that we've been writing up. And so that is the extent of this work to this point. And I'd like to thank all of the collaborators that I've worked with at the Danforth, because this has been very interdisciplinary.
Thank you so much for this presentation. So let's see, we have one question. Some compounds can only be seen in positive or negative mode. Can you comment on combining positive and negative mode data?
Yes. So we have worked with polarity switching. So we should detect compounds that will show up both in the positive and both in the negative mode. I think so we're not going to be limited by positive or negative mode. And we will also be working with Hillock data and reverse phase data in both of those modes.
Okay. Well, thank you very much. So let's see if we have more questions. no additional questions for the moment. So I propose that we take a short break to have some refreshments. I wish we could offer you refreshments. We can. So let's have a just virtual break, and everybody hopefully can stretch their legs and so on. And let us come back at 12:45. So a 10-minute break. And I want to thank all the presenters so far for staying relatively on time. I didn't have to bring out my big phone alarm. So it can happen in the second part, hopefully not. So 12:45 Eastern or 45 after the hour in your time zone. We stay at the same link, so please don't go away. We have 6 additional presentations, which look very, very interesting. So I look forward to that. And as Lauren is saying on the chat, thank you all the panelists for the great talks so far. Thank you guys. Okay, so 45 minutes past the hour, we will be back. Okay, and we are now back. Welcome back, everybody. Thank you for staying around. So our next speaker is Pavel Shliaha from University of Southern Denmark. And Pavel will talk about systematic optimization of protein sequencing for top-down proteomics. I look forward to hearing about that.
Okay, let me share screen. Okay, can you see my screen?
Yes, all is good.
Okay. Right. So, my name is Pavel. I'm currently working at Memorial Sloan Kettering in New York. But I'm going to be talking about the work I have done in the University of Southern Denmark in Professor Ole Jensen group. And that is about systematic optimization of intact protein sequencing. So before I start, I wanted to say that the majority of the code was contributed by Sebastian Gieb. I asked him to join today, but he's a medical doctor and he's actually cannot because he's fighting the coronavirus epidemic in Germany. So to go back to my talk, so there is 2 ways in which you can do proteomics. So one of them is the more conventional one, bottom-up proteomics, where we take the protein and digest it into peptides, and then analyze the peptides and infer the protein quantities and presence from the peptide level data. And the second one is top-down proteomics, where we omit the digestion and just analyze the protein molecules intact. That takes care of the protein inference problem, but it's a much more technologically challenging approach. And one of the challenges is we need to get very high sequence coverage across the protein backbone. And the way to achieve that is actually to combine different modes of fragmentation. So fragment protein with different methods. And at the University of Southern Denmark, we had Illumas, which had 4 different methods of fragmentation. So 2 different methods were based on collision with the gas, the CAD, which is the performing the ion trap, low energy collisions, HTD, high energy collisions, where can be performed in the multipole. UVPD can be performed is protein fragmentation by irradiation with the laser. And finally, ETD, whereby a protein is fragmented by getting an electron from the ETD reagent. And for those people who have done ETD, you know that ETD often results in incomplete fragmentation because even though the bond's broken, the protein molecule stays together through non-covalent interactions. And the way to deal with that is to shake apart these parts of the molecule by additional round of CAD or HTD. And these combined techniques, when you first do ETD and then you do CAD or HTD, are called ET-CAD and ET-HTD. So there have been a lot of different studies for protein fragmentation. Here's a bunch of them. But the biggest problem with them was that they had an operator who sat in front of the mass spec and basically punched in each of the fragmentation methods one at a time, acquired the data, and then moved on to the next method. And of course, when you are doing this, you cannot check many different fragmentation conditions. You can go through maybe 10 or 20. So we actually wanted to do something different. We wanted to study protein fragmentation systematically by testing, hundreds or thousands of fragmentation conditions. And then we also needed to come up with tools to effectively visualize and analyze this data. And we chose R because I have a history with R, but also because it has some very nice visualization methods that I'm going to show later, like ggplot. So just to give you a feeling for what our experiments are. So for example, when we wanted to test the fragmentation of a particular protein, we wanted to test fragmentation of different charges for the protein, different amounts of the protein, which is the AGC targets. For ETD, we wanted to test different amounts of ETD reagent, and then we also wanted to test different fragmentations with different ETD reaction time, different collision energies for CAD and HCD, different VPD reaction times, and if you take all these conditions that are present on the screen and make the combinations of these parameters, you'll find that you get 1,872 fragmentation conditions. So clearly, you cannot sit in front of a computer and acquire them one by one. So one of the biggest challenges actually to testing this condition systematically is the thermal method structure on the Lumos instrument. These thermal method files, they are not like TXT files or XML files from other vendors. They're binaries. And you cannot just go into the method file and start, modifying the method file manually. So the only, but of course, we also didn't want to sit down in front of a computer and input them in one by one. So we contacted Thermo, and they actually have a tool called XML Method Changer. And XML Method Changer allows you to go into an existent method file and modify the settings there. And one of the modifications that it allows to do is it allows to copy and paste a fragmentation condition. So, and that's what we did. We basically took a very simple target method file and then just made many copies of that fragmentation condition. And then we went into individual one and changed the settings. And the way you change the settings and do all of these manipulations is you have to supply a separate XML file that contains all of these modifications you want to make to the file. So here is our total workflow. So we decide what parameters we want to test. Then we go to R, we make an XML file that contains the changes we want to make to the method file to make this very big method file with different fragmentation conditions. We take that method file, put it on the instrument. We directly infuse the protein in, fragment it at many different conditions. We get a raw file. And then the spectra will be, and then the raw file is, of course, charge state convoluted and not deisotoped. We charge state deconvolute and deisotoped. We use an extract node in Protein Discoverer. We get MZML file. And then we put this MZML file back into R. And in R, we do the following things. So we look at the charge state deconvoluted and deisotoped MS2 signals, and we assign them to protein fragments based on the mass accuracy. We investigate what fragment type we have generated. We try to understand how individual parameters, which parameters mattered most, which didn't matter that much. And we also, if you're interested in a particular bond in your protein, you can see which fragmentation parameters actually produce a fragment from that bond. So let me give you an example. So this is an example of our raw file after acquisition. So it's a 2-hour acquisition. And at each point of time, each spectrum is a fragmentation at one particular fragmentation condition. So for example, on the upper left, this is an ETD spectrum. And on the right, this is a CAD spectrum for the same protein, myoglobin. Okay, so and then once we have processed this data, we can start to ask some smart questions about the nature of fragmentation. So for example, we can ask we know that fragmentation generates these types of fragments A, B, C, and X, Y, Z. So we can ask, well, do different fragmentation methods generate different fragments? So, for example, we can ask, well, CAD and HDD, they are both based on collisions with the gas, but is there a difference in the distribution of fragments? And indeed there is. So CAD tends to generate more Y ions, and HDD tends to generate more B ions. But interestingly enough, HDD is also generating some A ions, which we didn't expect. Another question that people often ask is, we also want to understand what is whether our fragments have additional protons in them, because oftentimes when you fragment a protein or a peptide, it abstracts a proton from the during fragmentation from its corresponding fragment. So, for example, in our data, all the Z fragments had an extra proton that we didn't expect to be there based on the gas phase chemistry. And yeah, so this is like a overview of all CAD, HDD, ETD conditions. This one is a very interesting plot, and it's so this is generated in ggplot, and this is a very informationally rich plot. So yeah, so bear with me here. So each of the dots on this plot represents one fragmentation condition. The size of the dot represents the number of fragments that we managed to assign in this spectrum, and compared to the maximum and the best fragmentation condition. And the color represents the proportion of intensity that we managed in our MS2 spectrum that we managed to assign to the meaningful fragment. So the best fragmentation condition would be have the biggest dot, and it would be yellow. So it produces a lot of different fragments, and most of the intensity is associated with this fragment. Now, the coordinates of the point on this plot represent the fragmentation parameters. So within the panel, the coordinate represents the ETD reaction time, and on the x-axis, and on the y-axis, it represents the strength of supplemental activation. So, and the position of the panel itself is defined by the amount of protein injected here, amount of ETD reagent injected here, and also the type of supplemental activation. So, for example, if you look at this plot, at this point, this is AGC for protein is 1 is 6, AGC for ETD reagent target 1 is 7, ETD reaction time of 5 milliseconds, and then the HCD was used as the supplemental activation with collision energy 14. And then just by looking at this plot, you can already start to have a feeling of what works with fragmentation and what doesn't. For example, you can see that the HCD tends to over-fragment compared to CAD. Like if you compare this panel and this panel, or you can say, does the amount of ETD reagent matters if I compare these panels, and the answer is no. Or within a particular panel, you can ask, what is the best ETD reaction time? And it's somewhere between 5 and 10 milliseconds. So it kind of tells you which fragmentation parameters matter and which don't in general. And the final plot I want to show is if you're interested in a particular fragment within your protein. So we wanted to make this like a fragmentation map, where we say, okay, this bond is covered with these conditions, this bond is covered with different conditions. But of course, because we have all these types of fragments, A, B, C for the sequence that goes to N terminus, and X, Y, Z for the sequence that goes to C terminus, it's very complicated to visualize all of this with colors. So we basically just said, okay, all the fragments that go to the N terminus, A, B, C, we'll call N terminal, and all that goes to the other side, X, Y, Z, we will call C terminal. And if there is a bond that is covered by both, that goes in both directions, we just call it bidirectional. And then what you can do is you can do like a fragmentation map like this, where you can say, okay, this is a big table, and each column represents one fragmentation condition, and each row represents one bond. And then you can basically say, okay, if I'm interested in this bond, it's covered in CAD, in HCD, in ETD, in VCAD, but maybe not in ETHCD when the under this fragmentation condition. So we did a lot of more data analysis with this data, but I've just decided to show these ones because they are the easiest one to understand. Yeah.
Well, thank you very much for this very interesting presentation. I think this is a fascinating example of combining, R and experimentation to optimize future experimental designs. We don't have questions, but maybe they will come in a minute or at a later time point, but very, very interesting work. Thank you for sharing that. So let's give another minute if somebody wants to ask something or comment. Here you go. So Lawrence is very encouraging, says, thank you, Pavel. Thank you indeed. So, and it's really nice to also see how you used the R kind of functionalities with that. That's also very interesting. Okay, so in this case, let us go on to the next presentation. So our next speaker is Grzegorz Skoraczynski from University of Warsaw, Poland, and talking again about optimal transport for mass spectrometry. And the project is called Mass Spot.
Okay, can you hear me?
We can hear you and we can see you.
Okay. Do you see my screen?
And we see your screen. All is good.
Okay. Great. So, hello everyone. My name is Grzegorz Skoraczynski and I am from Faculty of Mathematics, Informatics and Mechanics of University of Warsaw. This is in Poland. And today my talk will be about the MassSpot, a Python tool for MSI clustering. What is the most interesting in this work is the method for comparing spectra, which is based on optimal transport theory. Probably most of you know how mass spectrometry imaging works. Let me introduce this briefly. Suppose we want to identify healthy or affected tissues in some histopathological material. Consider MALDI, which is matrix-assisted laser desorption ionization, and we take thin tissue slices, mount them on the microscope slides, and apply the MALDI matrix. The matrix is ionized with the laser and then heated, which causing subliming of this. Then we collect the spectra pixel by pixel and we get the picture. In every pixel of this picture is one spectrum. These spectra are clustered, for example, based on distribution of particular substances, which is done more or less manually, or some similarity measure like similarity index. In present work, we present the MassSpot, a Python 3 package for MSI clustering. Spectral similarity measure is based on optimal transport, and the method can take full spectra without any extra preparation except centroiding. Alternatively, you can only denoise somehow in order to improve the speed of computation. And how do we calculate the distance? Today, there was a presentation about optimal transport in different applications. Let me introduce this one more time. Suppose we have two spectra. These are isotope envelopes of similar peptides. There's a difference of 1 Dalton and also slight differences in peak distribution. And how does optimal transport work? Imagine that we want to transfer one spectrum into another. So we should move the appropriate amount of ion current from this point to this point, and from this point to this point, and from this point to this point, but leave small amount because there's a different distribution of peaks. So so we should move some part of this peak to this part and some part of this peak to this part. In general, the transport plan is the sum of amount of ion current times the distance to be moved. The optimal transport, or the Wasserstein distance, is the best, meaning the cheapest transport plan. So in general, imagine that there's a guy, let's call him room size, with excavator who is transforming one picture into the another. Imagine also that our spectra are some sand sculptures. The Wasserstein distance is also called Earth mover distance. When we think that we moved amount of ion current, it can be proportional to the weight of the sand with which we move. And the distance is proportional to the distance of sand which we, of the distance to move the unit of the sand. And then the optimal transport can be proportional to the work known from physics, which is the energy used to transform one picture to one sculpture to another sculpture. Of course, when we talk about optimal transport, we talk about infinitesimally small units of sand, so which and every unit can be differently transported. So also dome size has infinitesimally small excavator. Coming back to spectra, there is an algorithm, and there is a theorem which says that Wasserstein distance can be calculated in O of n when the n is sum of spectral length. Unfortunately, pure Wasserstein distance is not enough to solve all challenges. In present work, we propose the generalization of Wasserstein distance. Imagine the situation from this picture. One spectrum shows only several peaks of substances of interest. They may be simulated using some software or another way. And the other one is noised experimental spectrum. The Wasserstein distance will try to pack every one of these noised peaks into this spectrum. And this is not what we want. So maybe we want to move the noise to the trash or the dump truck, which can go somewhere outside the slide. We can do this with some constant penalty coefficient and proportionally to amount of dumped ion current. Imagine the other situation. These are two similar spectra of two similar peptides, but they are differently noised. So maybe we want firstly to remove the noise to the dump truck with some constant penalty, then transport optimally what is to be transported, and then fill the noise from the other dump truck with some constant penalty. Summing up everything what I said, the generalized this Wasserstein distance of two spectra mu and nu is the sum of optimal transport between mu and nu and the penalty coefficient lambda times the sum of moved to the truck noise and noise field from the other truck. So we can see that our measure is, for example, symmetric. In reality, it is symmetric because potentially the triangle inequality may be not satisfied. Let's talk more about our generalization. Our generalized Wasserstein distance is stabilized by regularization by Shannon entropy and total variance. So if we are transporting the ion current and there is a small perturbation in isotopic envelope, we don't know why. For example, from some problems with measurement or whatever reasons, we do not bother with this. The stabilization accepts to stick to these perturbations without any cost. So coming back to the building ideas, there's a small bulldozer or earth mover which can easily refine everything we need. So our generalized Wasserstein distance has some pros. For example, stabilization, as I said, distance is bound. And this gives robustness to potential artifacts in spectrum. Even multi-dimensional spectra can be compared because this method is so general. But there's one problem. It results in high computation complexity. But we implemented an iterative method which uses sinkhole algorithm and was presented by these guys from France. And do you want to see an example? there's a one. Imagine an image we obtained from metabolites, and this is the image of mouse cerebellum, which is obtained using MALDI orbit trap, and this is 21 to 81 pixels two-dimensional picture. We perform peak picking and put the picture into our package, and this is the pictorial result of clusterization using Masspot package. The result sticks to the original predictions of authors from original paper, and they see here even white matter and granular layer, but please do not require from me to see with them. We also plan to perform some further research, for example, find some other applications for bigger molecules, or we are trying to apply this method to other instrumental methods like NMR. And also we plan to implement further improvements and optimization, for example, better memory usage during calculation of this distance and so on. If you want to ask offline, you can contact me with this email, skoraczynski@mimov.edu.pl, and this package is publicly available at GitHub. And thank you for your attention. And do you have any questions?
Thank you so very much. Thank you. I loved the construction worker analogy. That was wonderful. Kylie is saying very cool. So very nice. And there is a question. Have you compared Wasserstein distance with dot product algorithm?
Yes, we do. And results stick to this. Also, we performed some analysis with dot with this similarity measure.
Can you say a couple more words about your implementation? I probably missed that. So how is it implemented right now? And a related question, could you show the GitHub URL again?
Of course. GRZ like Grzegorz, SKO like Skoraczynski, and Masspot. This is implemented in C++. We used the eigen library for calculating heavy matrix to do heavy matrix computation. There was the decision to obtain the package which is able to run even on poorly configured servers where there are problems with LAPACK. And this is and we implemented this synchro algorithm, but unfortunately it is too complicated to give a very short introduction to this because it's not for so short presentation.
Well, thank you very much. So if any of the attendees or panelists have questions, I very much encourage you to contact Grzegorz maybe offline or towards the end of our meeting. So thank you again. And so let us move on to the next presentation. So our next speaker is Javier Gomez from Vanderbilt University in the US. And Javier will talk about tools for leveraging high-resolution mass spectrometry-based detection of stable isotopes for metabolomics. And we can see your screen. And you can start anytime.
Okay, can you hear me?
Yes.
Awesome. Hello, everybody. As Dr. Olga mentioned, my name is Javier Gomez. I'm a PhD student at Young's lab at Vanderbilt University, and today I would like to talk about my project title, Tools for Leveraging High-Resolution MS Detection of Stable Isotopes for Metabolomics. So, in our lab, we focus on experiments using stable isotope labeling as it gives us information to convey metabolic flux analysis. In order to do that, we feed labeled molecules to different organisms, and depending on the molecular structure and the labeling of the molecule, the metabolic products will get labeled in defined patterns that can be analyzed using GCMS or LCMS. The labeling of the products, however, is not immediate, and most of the species get labeled at different rates. So if we analyze the labeling at different times, we can map the fluxes of the metabolic network, giving us an idea of how fast the metabolite is converting to its products. Usually the fluxes are shown in metabolic maps, as I'm showing in the figure in the right, where the width of the arrows are proportional to the fluxes. When we started working on our experiments, one of the first problems we realized was that most of the publicly available tools do not update their algorithms according to the latest advances in technology of the equipment. And specifically, over the last years, the resolution of mass spectrometers has been improved so much that we're able to resolve the mass defect between labeled species such as 13C and 15N. And currently, part of the metabolomic community is migrating towards the use of multiple stable isotopes in the same samples. For example, a research team in Mainz fed E. coli with media labeled with 13C and 15N and discovered new modifications in the RNA pathway, helping to better understand the metabolism of the organism and the molecular pathways associated to it. The study was possible because it was a targeted experiment where the researchers knew a priori the retention times and the molecular weights of the nucleosides when pre-processing and processing the LCMS data. However, in an untargeted study where all the retention times and the molecular weights of the labeled species are unknown, these types of experiments become quite impossible. The closest available tool that can handle a similar problem is X13CMS, that uses the data from a wild-type group and a modified organisms, both cultured in labeled and unlabeled media. And by comparison of the mass shifts, it is capable of detecting features or molecules that show differential labeling. However, when we try to analyze samples that were labeled with both 13C and 15N, the software didn't yield good results, as it could not handle information of different times, and it could not be linked to our metabolic flux analysis. With that being said, it is our objective to create a tool that can leverage the information given by multiplex high-resolution LCMS metabolomics experiment, focusing on the detection of labeled metabolites. So going into the details of our current algorithm, it starts with a list of all the features and the information of retention time, mass intensity that's provided by pre-processing the data with XCMS. It is also required to input the resolution of the mass spectrometer, the retention time window for coeluting compounds, and a vector of the time points of the experiment and the atoms that were labeled. Currently, the tool only takes into account the most popular labeled atoms, which would be deuterium-2H, 13C, 15N, and 18O. It will then consecutively take each peak, assume it is a monoisotopic mass, and find all the possible isotopologues in the list of features that are within a retention time window and that have a maximum mass shift. Once the list of all the isotopologues is created, it is saved as a bin. The tool will also search for possible adducts within a pre-made list based on the most common adducts found by the Phen lab. With the list of bins, and for each one, a vector containing the information of the intensity behavior over time is created, and the bins that do not show a labeling pattern are discarded. The remaining bins are fit to a mathematical model to extract the labeling speed information. And finally, the software uses a k-means clustering algorithm to sort the metabolites into 3 clusters based on the labeling speed. And we would expect to sort all the metabolites in fast, medium, or slow labeling. So in order to test the tool, we conducted labeling experiments in plant and animal cells. To test the binning and filtering part of the algorithm, we used 2 data sets. The first one on Arabidopsis roots with 13C5 and 15N2 dual labeled glutamine focused on the TCA cycle metabolites. And another data set that consisted of camelina seeds with uniformly 13C6 labeled glucose, where the metabolites of interest were the lipids that were produced. So after implementing the code to the .csv file that's provided by XCMS, a .xls file is created with 2 sheets. The first one contains information of all the bin features where the mass, retention time, bin number, and the number of labeled hydrogen, carbon, nitrogen, or oxygen atoms of each feature are shown. And the second sheet contains information about the bins, the number of labeled atoms, the retention time, and the monoisotopic mass for each one of them. It is interesting for us that the first data set started with 8.3 thousand peaks that were reduced to 512 bins, and only 23 of those bins showed a labeling pattern and were categorized as labeled metabolites. In the second case, in the case of the second data set, it started with 28,000 peaks that were reduced to almost 2,000 bins, among which only 88 of them were categorized as labeled metabolites. So generally speaking, that means that approximately 2% of the features that are resolved by XCMS correspond to actual labeled metabolites. In order to try the capability of clustering of our algorithm, we used another data set coming from a murine cell line of pancreatic beta cells cultured with uniformly labeled 13C5 glucose, focusing on the TCA cycle. And the reason why we changed the data set for this part is because our lab had already analyzed the data set in a targeted way and had conducted a metabolic flux analysis, giving us a better reference to compare. After running the algorithm, 8 metabolites were categorized as labeled, and they were identified compared to an in-house library. So we found that 6 of them were part of the TCA cycle, and more interestingly, the metabolites that lied closer to the input, in this case, glucose that was converted into pyruvate via the glycolysis pathway, were clustered as faster labeling, whereas metabolites further from the input in the metabolic network were clustered as slow labeled. So in conclusion, we acknowledge that the data processing and analysis remains a bottleneck in a normal metabolomics experiment when stable isotopes are used, especially if the study is an untargeted study. We also realize that most of the information, and that's about 98% of the features that are resolved by XCMS, or that we gather from a high resolution LCMS run, is actually not relevant to an experiment, being most of the features just either noise or adducts or unlabeled metabolites. And finally, we think that transient labeling can give enough information to reconstruct metabolic networks and improve the pathway analysis. So for our future work, we aim to implement the algorithm in a C++ based standalone tool that can run the whole process, starting from the raw files, leveraging the tools from OpenMS, and giving information to both targeted and untargeted experiments. We also want to see if we can use the information from the transient labeling to build metabolic networks de novo. And finally, I would like to thank the NIH for the funding and my team, Drs. Allen, Evans, Young, Kamapati at Vanderbilt University, also Jay Holman, and the Donald Danforth Plant Science Center, and everybody here for your attention. And I would love to answer your questions. Thank you.
Thank you so very much. So let's give it a couple of moments to see if we have questions or suggestions or encouragements. All is welcome. And if not now, I'm sure that it will be appropriate to ask questions at a later time, right? Or reach out to you directly. Please don't hesitate to do that. So since we don't have questions, thank you very much again. So since we don't have questions, so we will go ahead and move on to the next speaker. So our next speaker is Kavya Sharman, also from Vanderbilt University in the US. And Kavya will speak about workflow for deep statistical multivariate analysis for spatially targeted proteomics data.
All right, thank you. Just double checking you can hear me and see my screen.
We can hear you and we can see you.
Amazing. Hi, everyone. My name is Kavya Sharman, and I am a graduate student in the Caprioli Laboratory at Vanderbilt. And today I want to show a brief story that really was the result of a lot of interesting multidisciplinary work. And my background is primarily computation in the lab, but we also have a lot of great mass spectrometrists, experimentalists, and immunologists. And so I'm really excited to tell you a story that kind of came out of this really interesting collaboration. So before I get to the workflow, just as a really large overview, I probably don't have to tell you that staph aureus is a very large public health concern. Not only that, but it's also a very costly concern. So we have over $20 billion per year that are spent on excess healthcare costs, 35 billion in societal costs, and 8 million additional hospital days per year. Additionally, over 19,000 people die from this. And so it's a very serious concern. So a little bit about staph. It's a gram-negative bacteria. And a really interesting hallmark of a staph infection is the building of abscess. And so shown here on the right is a PAS stain. In the center, you can see the middle, this is the abscess, the actual bacteria. And on the sides, you can see kind of the leading edge as the bacteria kind of expands into the host. So a little bit more about this host pathogen interface. Shown on the left is another example. So this is a kidney, but in this case, it's a fluorescently labeled kidney. And so you can see the fluorescently tagged staph. And what I'd like to highlight is the abscess community and the leading edge that's kind of shown here on the right. And so, as well as the uninfected cortex. And this will be important kind of as I talk about the experimental workflow. But the real major takeaway from this slide is the host pathogen interface is really complicated. So right at that kind of leading edge, you have a lot of complex interplay of molecules where you've got macrophage recruitment, neutrophil recruitment, epithelial cells from the host side, as well as a lot of microbial proteins. So you can see some different microbial metal scavengers. And so it's a very interesting interplay. And the question that we wanted to ask in this study was, how do we really study the complex proteome while maintaining spatial integrity? So in order to do this, recently our lab came up with the method known as microLISA, and this is a method for spatially targeted proteomics. And so within this workflow, I'll walk through it fairly quickly, but we take a mouse and retroorbitally inoculate it with a fluorescently tagged staph strain. From here, we excise the organ, cut some cryosections of these. These are approximately 12 microns in size, and then thaw mount them onto a glass slide. From here, we can do some fluorescence microscopy and generate images such as the one you see here on the right, and basically identify regions of abscess, leading edge, and uninfected cortex that we would want to target for proteomics. From here, we take that cryosection and do a targeted trypsin application. And what I'd like to highlight here, the kind of novelty of this experimental workflow, is that we can use a piezoelectric spotter to deposit picoliters of trypsin at a time. And so you end up with spot sizes of approximately 120 microns in diameter, which is a lot smaller than kind of your standard LISA workflow. So from there, we do an incubation and then use ELISA to extract these tryptic peptides and then process them using a standard LCMS workflow. In this particular work, we used an Orbitrap fusion at 120,000 resolving power with a mass range of 400 to 1600. And I'd like to invite you to see Daniel Ryan's paper in analytical chemistry that came out last year for more detailed methods. So once we have this proteomics data, we use MaxQuant for some processing and generate intensity values for proteins. So here it becomes an interesting computational question. So how do we study this very kind of unique proteomics data set? And the kind of goal of my work, our work here, was to develop an unsupervised exploratory method to assess this proteomics data set with the following characteristics. It's spatially targeted. We have 3 regions, and specifically with this work, we had 2 time points. So we have 4 and 10 days post-infection because we wanted to see how is this proteins changing over time and space. And it's also a highly dimensional data set. So we have 39 samples, but over 3,500 proteins that we're getting measurements on. And there's also a lot of missing data. And so the reason that that's important, that I want to highlight it, is that standard proteomics approaches that are very robust typically involve things like differential analysis, which is very difficult when you have a lot of missing values. And we didn't want to sacrifice the integrity of the data by doing a lot of imputation. So the method that we developed involves a PCA coupled with the k-means, and then we do some network analysis at the very end. So I'll walk you through some of that today. So when we did the PCA, what we can immediately see is that the first 2 principal components that comprise about 30% of the explained variance can do a really good job of separating the data. And you can see the cortex, the abscess, and the leading edge here. The other thing that I want to highlight is that I've also shown time points. And so the large circles represent 10 days post-infection, and the small circles are 4 days post-infection. So we then took this data and did a k-means clustering. But in order to do the k-means, we first did an analysis by testing different k's, like different values for k. So we tested k from 2 to 15, and we determined 3 to be the most optimal using silhouette scores as a metric. And this is also biologically interesting because, as I've shown before, we had 3 regions. So it's really interesting that the data was able to kind of cluster unsupervised. And what I'd like to show here is that not only did it cluster, but one thing that seemed at least off for me when I first saw this data was that there were some leading edge samples that were actually clustered as abscess. But when you go back and you look at the time points, what you can see is the ones that were clustered as abscess were actually 10 days post-infection leading edge. And so this is a really interesting biological story, because what we're seeing is that with an unsupervised approach, just looking at the proteins that we're seeing, the proteomic signature that we're seeing at 10 days post-infection in the leading edge is starting to look remarkably like the abscess or the center of the infection. So we wanted to take this a step further and say, okay, what do these protein fingerprints really look like? And so this is a very qualitative approach. On the x-axis, we have the proteins, 3,500, and then on the y-axis, the average centroid. And so shown here is the protein signature for the cortex, the abscess, and the leading edge. And when you overlay these on top of each other, you can see that there's very little regions of overlap, indicating that these show differences within the protein signatures per region. So from here, what we can do is do a little bit of a biological characterization. So here we subtracted the cluster average from each cluster and selected the top proteins for network analysis. And here I'm showing the results from string. So what I'd really just like to point out is that as we move from uninfected cortex into leading edge, and then finally into abscess, we start seeing an increase in the number of immune-associated biological responses. And so initially, we have a lot of metabolic processes in kind of the uninfected normal region of the kidney. And then as we move on into leading edge, you start seeing some acute phase response. And then finally, by the time we get to the abscess, it's a full-blown immune response that we're seeing. And so in conclusion, just wanted to show that really we developed an unsupervised method for building protein fingerprints for spatially targeted proteomics data with high dimensionality. And the kind of key thing that I hope you take away from this is that we had a very complex data set with a lot of missing values, a lot of dimensionality, and using this unsupervised method, we were able to assess kind of how these protein fingerprints, or how all of these proteins are changing kind of in concert over time and across space. Further within this work, we applied microLISA and this computational workflow to investigate infectious disease and abscess morphology. And finally, within a staph-infected kidney, we used an unsupervised approach that I talked about today to identify immune response pathways that are associated with the host pathogen interface. And with that, I'd like to thank Drs. Caprioli, Spraggins, Vandeplas, and Scar, as well as my collaborators such as Emma, Danielle, Elizabeth, Heath, and Will. And thank you all for listening. Thank you, Mina and Olga, for organizing this. And I'd love to take any questions.
Thank you so very much. I am really delighted to see so many presentations that focus on spatial distribution as well. Quite a few questions. Good talk. How does the script handle missing data?
So yeah, a good question. So right now the output is from like the max quant intensity data. And so we've just left all of the missing data to be as zeros. And then because the k-means really doesn't handle that well, what we did on the front end was to do the PCA for dimensionality reduction, as well as capturing that variance. And then from there, we were able to run the k-means on top of that.
And so a few questions related to PCA. What was the percentage of variation along the third principal component? Any resolution for time along the other principal components?
Good question. So I don't have that figure shown here. This was just kind of a quick overview for the purpose of this talk. So the third principal component, I'm trying to remember. So we had 22% for the first, 7% for the second. And by the time we got to the third, it accounted for about 4% of the variance.
And another clarification question. Can you say again how you dealt with missing values prior to PCA?
Yes, so we imputed a 0 for any proteins that were not detected.
And one more. The distribution of the points in the PCA plot suggests that the clusters, after applying k-means, are somehow sensitive to the linkage method of choice. Have you checked this?
Linkage method. I'm not sure I understand the question. So we used the PCA, the correlation matrix. And so I'm not sure if you could maybe rephrase that and I could answer that offline.
So I think the question related to the distances. So when you do k-means, so you assign each point to the closest cluster. And so it depends on the definition what it means to be the closest cluster.
Oh, absolutely. So yes. So yeah, the more, we didn't use a Euclidean distance, we used a cosine distance. And the reason for this is that we didn't want the height of the intensity, if you will, to really drive the model. We wanted more of the angular cosine distance.
And the distance was defined with respect to the center of the cluster or the furthest neighbor? How?
The center of the cluster.
The center of the cluster. Okay. I'm personally wondering if some of the data structures and methods in Cardinal can be useful for that, because Cardinal doesn't have to work with raw spectra. So if the data come in form of a data cube, a lot of the methods in Cardinal are applicable directly. So I'm just curious if that can be an interesting alternative for you.
I think so, absolutely. This was very early on, like work, but absolutely, I think Cardinal, and then I enjoyed the talk on MS stats, and I wonder if even that would be an interesting approach to take. I'm more than happy to have more conversations, and also I'm always open for suggestions and how we can make this work better.
That sounds great. We've never actually combined, the MS stats side of research with the Cardinal side of research. So this may be an interesting opportunity indeed.
Hi.
Well, thank you very much for the great presentation. It looks like we do not have more questions. And if anybody has questions later, please don't hesitate, or suggestions or encouragements, please don't hesitate to add. And otherwise, we're going on to the next presentation by Michał Startek from University of Warsaw, Poland. So we have a lot of people from Poland well represented here, which makes me very happy. And so the topic of the presentation is IsoSpec, a high-performance calculator for high and full resolution isotopic spectra.
Okay, so thank you for the introduction, and I'm going to share my screen now.
And maybe your video as well.
And the video, okay, give me a second. Now that I've shared the screen. Oh, there it is. Okay, let me switch to the presentation.
Here you go. Perfect.
Okay, so let's start. IsoSpec is a simulator of theoretical mass spectra. It uses the concept of measure concentration, which I'm going to explain in much more detail a bit later. The main point of this library is that it's written in C++ with very hard emphasis on speed and computational efficiency, both from kind of C++ like micro optimizations and from algorithmical point of view. So it's several orders of magnitude faster than the previous software used for infinite resolution mass spectrum simulation. The library is written in C++, but it can be used also in C, Python, and R programming languages. Of course, each of these programming languages with their own caveats. So if you're really interested in very high speed, you probably shouldn't be using R. At least that's my experience. The library is mostly cross-platform, so it works on most common operating systems. It's free, open source with a very permissive BSD license. It's on GitHub. The link will be shown on later, so let me move on. One important thing to note is that it is a software library, not a graphical user interface program, so you kind of have to program to use it. You can't use it from as a standalone application. So I guess this presentation is aimed more at developers of mass spectrometry software, not like chemists. Okay, our users, here is the list of our users. The bottom two are my friends. In fact, the Wasserstein MS program was presented a bit earlier. Okay, so what's the deal with measure concentration? The problem with theoretical infinite resolution spectra is that they are very complicated, and they usually contain a lot of configurations that are very, very unlikely and will not be seen in practice in an actual spectrum produced by an instrument. Basically, what we usually are really interested in is the set of configurations which covers, for example, 99% of the total probability of ions. And this set is smaller by many, many orders of magnitude in most cases. And because of that, it's actually workable, because the full spectrum is often on the order of 10 to the 20 configurations or so. So it's impossible to see. Okay, so I planned this as a more of an hands-on kind of presentation. So let's see it in practice. I will be using Python, as that's what I'm most familiar with. So isospec is available on, okay, maybe let's do it like that. So let's construct a simple spectrum. So let's say we want to produce a spectrum that contains all configurations above a given probability threshold. You use the isothreshold class for this. Let's actually produce a full spectrum. So as all probabilities are non-negative, inputting -1.0 as the threshold is going to produce all of them. And let's start with something simple like C100, H100, which of course doesn't make chemical sense, but just an example. Okay, so here's our spectrum. And just to show you how much is it going to be, it's 10,000 configurations, even for a very simple molecule like that. If instead we compute just the 99% of the same molecule, and I didn't save it. Let's call it P. The length of P is just 7, and you can plot it. It's going to look like that, pretty much. I guess what everyone has already been used to seeing. Okay, so let's try something different. Let's import a module with several chemical formulas just for testing, and let's use the averaging molecule of size, let's say 2000 amino acid. We're going to multiply this by the molecular weight of the averaging, because this accepts the target mass in Dalton. So that's our formula. And let's produce a spectrum of 99% of that formula. As you can see, okay, I again didn't assign it to a variable. Let's call it A. Okay, so as you can see, that is 0.15 billion of configurations. No, it's 15 million of configurations. And it was computed in half of a second, so it's very fast. you can, I won't attempt to plot it with matplotlib because it's going to choke on it, but if I bin it, let's say with a resolution of 0.30 Daltons, this is a little bit slower because it's mostly used for debugging. You can see that the plot is going to look like something like that. Let's, for example, zoom onto this peak. And you can see the precise isotopic structure of each cluster with the same nominal mass. So obviously that's going to be useful for spectra with very high resolution. And there is also some more functionality, like you can do basic operations on the spectra. You can normalize the spectra. You can add them, and so on. But I guess I'm going to running close to the time limit, so I think I will just. It's best that I should. Yeah, switch back to the thank you page and accept questions. Okay, so here's the GitHub link again. If anyone wants to try it, you can contact me on this email. And thank you for your attention.
Thank you very much. This appears to be a very useful tool. I'm hoping that somebody has questions among the audience, so please don't be shy. We'll give it a couple minutes. So let's see, we do have questions. Timo is saying, very nice talk. Did you already have a look at the neutron star preprint?
Yes. One thing I do have to notice that neutron star is optimized with molecules for the use case where the molecules consist of elements in which the element has a very complicated isotopic pattern, which is very much not the case with biomolecules and large biopolymers. Most elements where the molecule consists of elements like carbon, hydrogen, some sulfur thrown in. I suspect, I think, is going to be faster than neutron star, which is like regular use cases. You can see in their preprint that they're testing on toy molecules like xenon 50, which and the only protein they test on with like sensible probability thresholds, like I don't think people are going to be, there's much use case for revealing 10% of the spectrum. In these use cases, even their preprint shows that I suspect is faster.
Well, thank you for the insights.
But I have to test it further. Okay, thank you.
Timo says thank you. Kylie is asking, are the R bindings as complete as the Python bindings appear to be?
Sadly, no. Basically, Python bindings expose also the generator kind of. Okay, what I presented, all of those bindings, all of those methods would produce the isotopic envelope and store it in memory. So it's kind of non-lazy computation. In C++ and Python, we also expose kind of generator or iterator, call it, using the C++ terminology, where you don't actually store the entire envelope in memory, which is really non-feasible for molecules like titin or something or the very, very extreme proteins. We don't expose these in R because in my experience, R just isn't, if someone is interested in that kind of high performance computation, he's going to be probably using C++ anyway.
And a related question, okay. So Kylie continues, sounds like R6 or reference classes could be useful.
R6, I don't know what that is. And in fact, I would have to ask Mateusz about those. But yeah, thank you. We'll have a look into it.
So it looks like you guys need to talk, right? So that
yeah,
seems to be...
I'm the Python guy. He's the R guy.
That's right. Okay. So another attendee is asking, where can we find user-friendly manual?
A little bit in the future, sadly. It is a very, very beta. What I presented is very, very beta, but we hope to have it like nice and polished in time for ASMS. The pandemic got a little in the way of getting it ready for Boston, but hopefully not more than a month away.
Sounds good. And one more question. Can this be used to generate peptide spectra given a amino acid sequence?
Yes, I mean, obviously you have to translate FASTA into a chemical formula. And this is, we have already done this in C++. We still need to expose it in Python. It's going to be in the next version, like 2.1.0, which I showed the pre-alpha version of. It's not ready yet, it's in works. But right now you can just convert the FASTA to chemical formula yourself. It's not very hard.
Thank you very much. So let's see. How about C++? Oh, sorry, C#.
We don't have bindings for that. I guess C#, I think C# makes it very easy to use functions from C, and we do have an interface in C, so you can use the C++ algorithm exposed by C and in C#, but again, the last time I touched C# was like 7 years ago, I think, so I may be out of date. Maybe they can call C++ easily right now as well.
So it looks like Kylie has a comment. So Kylie, why wouldn't you just use your microphone? Maybe it will be easier.
Oh, I was just commenting that R6 and reference classes are just a mutable object-oriented style for R, more similar to the Python or C++ style classes. Useful for things like iterators and anything with mutable state if that's what you need.
Okay, I'll have a look into them.
Okay, thank you very much. It looks like this work has a lot of potential, and I'm really excited to see how it will evolve later, and hopefully in interaction with some of the people in this group.
Okay.
So now, thank you very much. So last but not least, we have a presentation by Erick Velasquez from University of California, Los Angeles. And Erick will speak about targeted MSQC, an R package for quality control and peak verification in targeted proteomics.
Hi, thank you everyone, and thank you for the introduction. Can everyone see my screen?
We can.
I didn't have the time to test it. I mean, it was like...
Okay, so today I'm going to be talking about automated quality assessment of chromatographic peaks and targeted mass spectrometry data using machine learning. And I'm a graduate student at UCLA, but this work really stems from an internship that I did at Genentech. And so at Genentech, we specifically work on biomarker development. And so just to give you a roadmap of like what raw data looks like into biological insight, specifically for biomarker development, it starts with data generation, then some sort of visualization, then doing quality control, and then afterwards doing data formatting, carrying it out to data analysis and then to interpretation. I'm going to be focusing specifically on the quality control aspect of it for a clinical design or for a study design. And so for quality assessment of targeted MS data, right now it currently requires a large amount of manual inspection of peaks. And so it involves human intervention in which an operator goes through and looks to see if peaks are correct or if they're usable or if they're not usable. And as you can assume, this can be time consuming and unscalable. It's prone to inter-operator variability. So within the different operators, there's different conclusions and also intra-operator variability. So maybe the same operator, but at different times has different conclusions. And it also creates operational challenges if trying to scale it to a large proteomic experiment. And so the automation of this would be very helpful. And the way that we're planning about doing this is that we calculate quality metrics and then build predictive models that then are able to assess the quality of these peaks and then do this in an automated process. And so we built an application or a package that we call targeted MSQC. And basically you want to use this package if you're trying to automate the flagging of low quality or misintegrated peaks, if you have large sample sizes, and then also we've included functions in the package to be able to integrate it within Skyline. And it's open source on GitHub if you follow the link. And the basic idea behind it is that we use machine learning in order to separate it out into different classes. And we've done a large amount of feature engineering in order to be able to essentially define what are some features that might be predictive of the quality of peaks. And so just to give you a little bit of an idea of what these features might be, I've outlined them here. I'm not going to go through every single one of them just because of purposes of time. But to give you an idea of what I'm talking about, let's take jaggedness, for example. We're using calculus in order to define what jaggedness is. And so when you look at a peak and then you take the derivative of this peak, it has certain inflection points. The more inflection points a peak has, the more jagged it might be, and therefore we can create a relative score around what the jaggedness of a peak is for certain runs. And we do this throughout for different types of features. And so the pipeline involves both an unsupervised and a supervised aspect of it that can be used jointly or independently. And so I'm going to go through the unsupervised pipeline first. Basically what it involves is that you do clustering and anomaly detection, and then this creates predicted annotations from the uncategorized transitions. These predicted annotations you can then take as your predicted results if you're interested in, for example, what are the best peaks and what are the worst peaks, or you can combine it with the supervised pipeline and use that as your training set. Alternatively, you can also create manual annotations within a supervised pipeline and then use that as your training set if you don't want to use the unsupervised version of it. And then from that, you create a validation and also you create and also validate a predictive model that then you can use in order to essentially render to the rest of your experiment to create predicted results. To give you an idea of what the supervised pipeline involves, you first annotate the training set within Skyline using the annotations features that Skyline provides. You then export the annotations along with chromatogram and peak information to targeted MSQC. You run the pipeline within R. You calculate the quality control metrics, build a predictive model, assess the model, and then get predictive results, which then you can bounce back into Skyline in order to be able to look at what the predictive results are giving you within the Skyline framework. And we've integrated different, along with the Carrot package, we've integrated different types of machine learning models that you can use. So for example, I'm giving you 3 here, decision trees, SVMs, or logistic regressions. And we've noticed that random forests tend to give you the best results with around 90% accuracy. To give you a visual representation of what this looks like, this is an experiment for a specific peptide in which we're doing it across multiple runs in measuring different transitions for multiple runs. And you can see here that, for example, this peak here carries out a large tailing, which might be due to interference between different transitions. And so that is picked out across replicates, across different transitions. And you can see here, for example, this one is completely jagged, and it also has a large retention time. But for example, within these peaks right here, you see that most of them are good or are consistent across light and heavy ratios, and then also carry out are well shape, and these would be something that you might be interested in quantifying. For the unsupervised pipeline, the workflow is much simpler. You export the annotations to TMSQC from a Skyline framework. You run the pipeline with an R, in which you calculate the quality control metrics, build the predictive model, and then use the predicted results and then visualize the results in in Skyline. And the basic idea behind the modeling is that we first do clustering in order to separate out into different classes based on the large variables that we've created, so all of the features. And then from that, we measure the distance of the distribution of each peak from the mean of the distribution through the Mahalanobis distance. And basically from that, we notice that points that carry out a high variance are usually things that are flagged, and things that have low variance are things that are okay. And so things or peaks that generally lie within the middle of the distribution or have low variance essentially are the ones that we would want to keep. And things that are very out there in terms of multidimensional space are things that we would want to flag. And we did this across 2 different data sets. And we noticed that it has around 90% accuracy. And we also carried out bootstrapping for this calculation. And so we carried out 10,000 simulations for each experiment. I wanted to give you a visual representation of what that looks like. So if we measure the Mahalanobis distance or carry out the scoring from it, you can see here that as the distance from the mean increases, you also get worse peaks. And so this would be something that you would want to quantify in terms that both the light and the heavy ratio seem to agree, whereas these carry out a large jaggedness and have a large dissimilarity between the light and the heavy ratio. And so in conclusion, targeted MSQC is an R package that can be integrated in large targeted mass spectrometry panels to predict quality of chromographic peaks. Unsupervised machine learning identifies chromographic peaks outliers and gives us a fast alternative to QC data without a training set. And the supervised machine learning automates the quality control according to metrics important for analysts in a reproducible manner. Give me 1 second. I'd like to thank the team that I work with at Genentech. And if you have any questions, feel free to take my personal email so we can carry out the discussion even further. So thank you.
Thank you so very much. This is very interesting, and we have a few questions. So let's see. Some just encouragements. Thank you, Erick. Wonderful talk. So this is great. Maybe the first question is, if you can say again about the supervised part of the approach, how did you train the supervised models? In particular, if you have some deviations from optimal performance, which are not part of your training set, so how do you make sure that the model is able to capture them in the future?
Sure. So we actually carried out manual annotations in order to train the initial supervised machine learning pipeline. And so we had an operator go through and essentially pick out, like, this is something that we'd want to use, this is something that we would not want to use. And we did this for about 1/10 of the data set in order to make sure that there was like no variability down the line. This is something that we're sort of starting to try to figure out now. And I think that leads us into the next question that they had about neuronal networks. And so something that we were trying to do was carry out transfer learning with deep learning in order to be able to first create sort of like a base model of what things might be good and things might not be bad. And then from that, extrapolate that to further or future experiments. And that's something that we're still working with right now and something that we're considering.
Yeah, so Erick is referring to questions which ask if neural networks were used. a related question to that, do you think you will have enough data to train neural networks in this context?
Yeah, that's a great question. We're hoping to get more data. This is something that we're just starting to work with now. And so we're hoping to not only do it for the clinical trials that we're working with at the moment, but also with other clinical trials across the organization. And then also just data in terms of like peak data from other publicly available data sets as well.
One more question. Can this approach provide recommendation for which transitions are interfering and should be removed to save the peptide measurements?
Yeah, so currently at the moment, it does not. I think that's a really good suggestion and something that we're working towards. For that, we would have to be able to cluster into even more classes, not just something that is like good or something that is bad, but something that tells us a little bit more about the shape of the cluster as opposed to retention time intensity. But that's like a really good suggestion and something that we're working towards.
My personal, hold on, before I make my suggestion, what is the difference between your package and mProfit?
So I haven't necessarily used mProfit as much, but from my understanding of it is that I think they do more heuristic modeling in order to be able to quantify a specific number of it, but I'm not necessarily sure. And so our package uses more machine learning models. And so the advantage of it is that you're able to tweak the model according to what it is that you're specifically looking at least for the supervised approach.
I think I maybe can help answer that. mProfit is not designed for QC. It is designed more for a quality of transitions for the endogenous samples that you acquire as you go. And I think your work is more on the standard samples, right, and reference samples.
Yes.
Right.
Yes,
and
yes.
So you're actually not working with samples that have actual biological variation, right? So you have the samples which are standards profiled in time, right?
Yeah. Yes.
That's right. And
Thank
so my
you.
personal suggestion was to maybe, I would be very curious to know how it relates to MS Stats QC, because MS Stats actually has a very extensive module which looks into similar issues. So I would be really curious to know, maybe not necessarily now, but at some point it would be really interesting to see how that compares.
Yeah, that's a really good suggestion and something that I'm definitely going to be looking at.
Okay, so in this case, if we do not have any more questions, this concludes our program for the May Institute. And so let me take this opportunity to thank once again everyone who contributed to making this happen. And so now on call, I see we have Kylie, we have Ting, and we have Mina, who have all worked very hard, and Lauren, one of our invited speakers, who all worked very hard on making this happen in a very short time. We literally came up with the whole plan in 2 weeks prior to the dates of the May Institute, and we had to be creative and quite flexible in the new format. And I think that this format worked as well as it could be expected. And so I'm very grateful to all the invited speakers. I'm very grateful to all the speakers who contributed their research today. I think this is a very important part of the program. And of course, I'm very grateful to all the participants who joined us at this time. I would like to have a special shout out to Mina, who did a lot of work in the background to organize all the logistic details, send the advertisements, handle all the registrations and all of that. So we are very grateful for everyone coming together in this time. So at this point, my biggest wish for you is to stay safe, stay healthy, try to be productive as much as the situation allows. And I very much hope to see you in person in one of the conferences that are coming up. And hopefully I will see some of you or some members of your group at the May Institute in 2021, which we do plan to make happen. So thank you so very much. Thank you to everyone who is not on the call with us right now. And I'm very glad that we had a chance to do it. Thank you very much.
Last one that I think is on behalf of all the people. Olga, thank you so much.
Thank you, Mina.
Thank you.
Thank you a lot.
Bye.
Well, hope to see you soon.